action remaining a perpetual possibility only in a world of speculation, creation, cellular division, nuclear envelope biogenesis. In biology, the single cell is the unit that we start with, and its process of division is quite important to inform and to have the correct division of the genetic material. So what we've based the last 25 years is on a particular compartment that contains the genetic information, and that is the nuclear envelope. What you see here in the figures is, in red is a membrane, which is the nuclear membrane, and during cell division, specifically in open mitosis, these at late anaphase to early telophase, these are words that we're going to be using throughout the talk, randomly and progressively. So during these various stages of cell division, what happens, there is a reformation of this nuclear membrane that is in red, and a completion in order for division to occur completely. So what is the nuclear membrane and its complexity? It is one of the more complicated membranes in the cell. It is a double bilayer, and it is composed of proteins and lipids, and its outer layer is interacting with another complicated compartment called the endoplasmic reticulum, where a lot of processes occur, such as protein synthesis and lipid synthesis. And the inner bilayer is also very important because that is the connection to the genetic material, and a lot of the processes that are required for regulation actually interact with the inner bilayer. We've studied this complicated membrane because if it's not formed correctly, what you can see in the lower panel there, this is the misformation of it. One of the pathologies that it ends up bringing about is early aging. So what you see here is this young fellow who's about, I believe, the age of about 8, who has the physiology of an 80 to 90-year-old man. And if you look at his skeletal aspects, this is how he is. This is basically, this is called, one of the pathologies, this is called during the laminipothesis, and it is progeria. So it is important, therefore, to have an understanding of this nuclear membrane to be able to understand how actually aging works. So we've spent a lot of time basically trying to understand, during the two processes of assembly, open mitosis, and the other process, which is around the formation of a new membrane around the sperm head during fertilization. There are two schools of thought that explain the nuclear membrane formation. One is the envelopment model, which occurs during open mitosis. And this model basically suggests that upon the formation of the nuclear membrane, there is an envelopment of the endoplasmic reticulum to actually form a new nuclear envelope with the incorporations of what you see as cylinders in purple of the nuclear pores. The other model, which is based on fusion, membrane fusion, is based on the fusion of what you see in red as vesicles that are in red. And these vesicles initiate the process of membrane fusion to actually give the formation of a completely new nuclear envelope, which is named as the male pronucleus. It is through this process of membrane fusion that we've had an understanding of what are the molecular mechanisms that have occurred for the formation of the nuclear membrane. To be able to study this, what we have also suggested during our investigations of the last 20-25 years is that these two models are not mutually exclusive, that they're actually related. And hence the membrane fusion occurring also in the envelopment model. To be able to go through this process of understanding the protein-lipid interactions and understand what could prevent the malformation of the nuclear envelope, we've begun with using simple biological models such as the sea urchin model. Then we've gone and used what we call a cell-free system and then gone into the cell. And to be able to actually understand what's happening in the cell, what we've been using is a multidisciplinary approach going from quantitative imaging, liquid and solid state NMR spectroscopy, nuclear magnetic resonance spectroscopy, lipid mass spectrometry, chemical biology tools, and advanced electron microscopy. So through the talk, you are going to see examples of these methodologies and why we've used them to be able to understand the process of nuclear membrane formation, and specifically initially the male pronuclear formation. So the cartoon events of the male pronuclear formation in the sea urchin, embryogenesis in vivo, is such that when you have the penetration of the sperm to the egg, the first thing that happens, nature's pretty crafty because the male component sheds its membrane, and it is the female component that actually brings in the new membranes to form the complete nuclear membrane. It is this complete formation of the nuclear membrane that fuses with the female pronucleus to start the initial stages of embryogenesis that goes into the cell division and processes into formation of the organism. So what we've done is that we've tried to imitate this process using what we call a cell-free system, and the imitation of that has been quite successful, and it has allowed us to be able to decipher each component of the process and understand how the mechanisms function. So what we do is that we spawn the sea urchins and we gather the eggs and the sperm, and we go through processes of centrifugation, basically separation of components of membrane versus soluble components, and further along we separate these population of membranes into populations that we can investigate. By adding these membranes into these various components in test tubes, what we can actually mimic is the formation of the male pronucleus in vitro. So that allows us to depict various components and understand what is actually occurring. So I've alluded to the phenomenon of membrane fusion. What do we mean by membrane fusion? So the nuclear membrane, as I had suggested, is complicated. It is composed, like most membranes, of proteins and lipids. And during the process of any communication in the cell, what occurs is that there have to be processes of membrane fusion occurring, which means that when you have two membranes at the vicinity of each other, you have a certain problem which is called water. And water has to be shifted out because the two oily surfaces, which are the lipid surfaces, have to get close to each other to be able to fuse into one another and to be able to pass on the information. So in order to be able to do this, one of the major components are a class of lipids, which here basically we can show this class of lipids are the phospholipids, and this is their head, and this is an inositol group, and this is their tails. And it's their tails which basically is what we call hydrophobic and is against water. These are the soluble components. When there is a particular reaction that occurs and there's the cleavage of the head via enzymatic processes that modify these type of phospholipids called the phosphoinositides, we obtain a type of neutral lipid called diacylglycerol, and this neutral lipid, in common terms, is headless. And because it's headless, its head has been cleaved, and you're only left with the fatty acid chains, it has a specific type of characteristic, and actually it is used in the process here to initiate membrane fusion. So it's very important that the localized process of membrane fusion occurs via this particular neutral lipid diacylglycerol. So if you remember the slide before, what I was suggesting is that while we go through the process of separating the membranes, what we got were different population of membranes that are responsible for the formation of the male pronucleus. Through many years of analysis and developing lipid mass spectrometry, what we showed is that a very specific membrane vesicle, which we call MV1, has a very unusual lipid composition. This unusual lipid composition is made out of these phospholipids, which have very charged head groups. So instead of the OHs that you see here, you have actually phosphate groups. This is very unusual in biology because classically these are in very small quantities, and they're actually known to be second messengers. So the messengers that carry various information from one part of the cell to the other via their specific proteins. But in this particular case, which was very unusual, we determined that this membrane vesicle has a very unusual property and is extremely charged. So what did that mean? The meaning of it in terms of membrane biophysics was studied using a method called solid-state NMR spectroscopy to understand the dynamics of such a charged membrane vesicle. Why is that important? Why is it important to study the dynamics of membranes? Because the proteins that are targeted to the membranes are actually very affected by the characteristic of the membranes. So whether a membrane is fluid, as the word says fluid, so it's disordered, or whether it's ordered and it's not fluid, is going to affect how the proteins interact. So again, it will affect how communication is processed through the cell. So what we did using deuterium solid-state NMR spectroscopy, we showed that this specific membrane vesicle, by doing different types of experiments called phase transition experiments, is actually quite fluid. Now, until these experiments were performed, people who worked in membrane biophysics, and specifically using phosphoinositides, couldn't have imagined that high levels of polyphosphoinositides existed, and they were stable, and also they were disordered. So this brought up the question that are lipid second messengers, such as the polyphosphoinositides and their derivative diacylglycerol, also regulators of endomembrane morphology and dynamics? So initially, to show this, we went through the process of, in the egg and during the process of fertilization, to label the vesicles that were containing this type of enzyme that modified the phosphoinositides, to see where they were actually localized. And what we determined was that in an unfertilized egg, these are actually different compartments to the complicated endoplasmic reticulum compartment, that they were a compartment by themselves. And during the 2 minutes of fertilization, when the sperm has penetrated the egg, what we saw is that there is an accumulation of these type of vesicles that are enriched in these phosphoinositides around the sperm head. And you can see the zoom here. So after 10 minutes of post-fertilization, we have the formation of the new nuclear membrane, the male pronucleus, but the initial steps is the fusogenic vesicles. So the vesicles that are responsible for the initiation of fusion actually are present in situ in the cell, and they were not based on the separation of our in vitro experiments. So this was quite exciting because we've shown a new compartment that existed, and we went a step further, and the step further was that what happens during the first division. And what happens during the first division is in the sea urchin, because it needs embryogenesis to go very quickly, it forms these organelles called carriomeres. So these are small nuclei that have their own nuclear membrane around them. And what hopefully you can see is that prior to the first division, again, these vesicles that are enriched with the phosphoinositides are accumulating to the regions where there is a necessity for these small nuclei to fuse together to form a bigger nucleus to go through the process of embryogenesis and division. So this sort of explained why there were so many vesicles, because every time there was a requirement for these vesicles to be present to initiate membrane fusion. So what we then found out through many other experiments is that the various counterparts, the proteins and the lipids that were being involved in this process of fusion were actually quite similar to the membrane fusion events that occurred during membrane trafficking in mammalian cells. So the same similar components were actually involved. So going back to the fusion model, we showed that the formation of the male pronucleus and further on the requirement of membrane fusion to go through the process of embryogenesis involved the proteins and the lipids that are present in membrane trafficking. So there was a parallel between the sea urchin system and the mammalian cells. So from that, we went through a more complicated system, and we moved on to the membrane dynamics in mammalian cells and asked the question is whether diacylglycerol, as a modulator of membrane dynamics and fusion, was also conserved in mammalian cells. Now, to be able to do this, we devised a new mindset, a chemical biology tool that would actually target the specific compartment under investigation, which was the nuclear membrane, and specifically also the endoplasmic reticulum, because the nuclear membrane is a subunit of the endoplasmic reticulum. So we hypothesized that if we use this specific tool where we could actually target these compartments and ask the question that with another part of this tool modify the diacylglycerol in such a way that we deplete diacylglycerol from those two compartments, would we still be able to have a new nuclear envelope formed during somatic division? So this is not during fertilization, this is not open mitosis that we are investigating now. So what you can see is the cell division that is occurring, and what you see in this high intensity images is that you have the formation of the nuclear membrane that you can visualize here. That is because you have not modified diacylglycerol, and therefore you have a normal division. Now, upon the localized modification of it, what happens is that in the modification process, when you target both of these compartment entities to the nuclear membrane, what you're forming is you give the diacylglycerol a head, and upon obtaining its head, what happens is that you no longer can see the nuclear membrane. And the endoplasmic reticulum has also lost its structure. So that was quite strange and good at the same time, because this showed that what we had seen in the sea urchin model, which is a model organism, actually paralleled in mammalian cells. But what was unusual was that the cell divided still without a proper nuclear membrane. So to be able to study the ultrastructure of this nuclear membrane, we went further and we used what we call correlative light and electron microscopy. So what does that mean? that means that we're correlating the actual fluorescence image to an electron microscope image of a single cell. This is not a trivial experiment to do. And what we did here is that we showed that these blobs that you see in fluorescence actually have an ultrastructure. They're these onion-shaped structures. There's a complete deformation of the endoplasmic reticulum. And not only that, is that what you see in red here is that the nuclear membrane is completely fragmented. And so that, here again, that without diacylglycerol, the nuclear membrane, the ultrastructure, also shows that there's a requirement of this one specific neutral lipid to be able to actually have a proper formation of the nuclear membrane during cell division. So what we then did is that we thought that what would happen if you put back small vesicles of diacylglycerol into the mammalian during the mammalian division, would we actually recover the nuclear membrane? And it's quite astounding, I'm still astounded by this data, is that when you add back at a specific time point in cell division the small vesicles of diacylglycerol, you get a complete reformation of the nuclear membrane. And the three-dimensional rendition of this experiment is as follows. So what you can see here is that without diacylglycerol, you have a completely fragmented nuclear membrane. And when you rescue the experiment with diacylglycerol, you have a formation of the nuclear membrane. And more positively, the cells went through their second division, and unlike not having a nuclear membrane, that they actually died after the first division, with the proper nuclear membrane, they went through division. So that meant that this particular lipid is quite important in the process of nuclear membrane formation, both in a system that deals with fertilization and during classical cellular division. So we tried the same type of experiments by microinjecting unfertilized eggs with the same type of modifying enzyme that actually put the head back onto diacylglycerol. And what we saw is that, again, if you can see, is that the more you affect diacylglycerol, the worse the egg becomes. Eventually, this egg exploded. So this is unfertilized egg. So that means that it's completely in a dormant form. And even in its dormant form, it still requires diacylglycerol to maintain its membrane morphology. The next step was to address the question of membrane vesicles. So are membrane vesicles actually involved in the process of membrane fusion? And here what we decided to do is to try and understand why in the sea urchin system that there's so many membrane vesicles, would there be the same type of membrane vesicles also in the process of membrane fusion? And if we look at, again, electron microscopy slides, what we can see is that here in the gaps of the nuclear membrane, we wanted to see whether there were any vesicles present or not. And what we can see is that there are vesicles present in the gaps of the nuclear membrane. However, we have to identify them, because the reality actually is that in one cell there are 14,000 vesicles. And how do we then take those 14,000 vesicles and actually characterize them to understand whether they are related to the MV1 type of vesicles? To do that, first we have to demonstrate that we're actually observing vesicles as opposed to tubules that are being transsectioned through. And for this, we used a type of electron microscopy that actually changes through the angle, and we can show that we're not observing vesicles, but we're actually observing, we're not observing tubules, but we're actually observing vesicles. So there are vesicles at the gaps, and the question is to be able to identify them. So the event is a vesicular fusion, we think, and to show that, we went a little bit further, and we used again our tool, our chemical biology tool, and here we targeted specifically these regions of the nuclear envelope that are very curved. And these regions of the curvature is where the pores actually are located. And we asked the question, is that if we modify using our tool the curvature of these regions, would we be able to actually visualize the incorporation of the nuclear pore complexes? So these were very challenging experiments to do and to quantify, and what we saw is that when we do modify, again, the presence of diacylglycerol, and I've done this complexity not for anybody to try and understand what the complexity is, but to try and appreciate that we can actually quantify each section of the presence of the nuclear pore and show that when you do not have, again, diacylglycerol, there is an enlargement of this area of the pore and a malincorporation of the nuclear pore complexes. So what I haven't told you is that the actual dimensions of these tubule curved regions are being affected by vesicles that come from another compartment that is partially associated and interacts with the endoplasmic reticulum, and that is the Golgi compartment. So the bottom line is that this is a very complex procedure. We think that membrane fusion is involved, and it is partially through vesicular fusion, and that diacylglycerol definitely plays a prominent role. So what we've shown through our investigations of the nuclear envelope formation is that diacylglycerol has two roles, not only a role as a second messenger, but it also has a role as a modulator of membrane dynamics, and also it has a role of localizing to extreme membrane curvature regions, which is required for membrane fusion. So it's not only the proteins that are responsible for membrane fusion, but it is actually a phenomenon that occurs between proteins and lipids. Time point. East Coker. To arrive where you are, to get from where you are not, you must go by a way wherein there is no ecstasy. In order to arrive at what you do not know, you must go by a way which is the way of ignorance. In order to possess what you do not possess, you must go by the way of disposition. In order to arrive at what you are not, you must go through the way in which you are not. And what you do not know is the only thing you know. And what you own is what you do not own. And where you are is where you are not. Preservation, molecular regulation. So again, our friends, the phosphoinositides, play quite an important role in the dynamics of proteins, a class of proteins which we call the AGC kinases, and we've spent quite a few years to understand in the cell, as opposed to in test tubes, how we can understand the dynamics and the interaction of two specific proteins, protein kinase B from this class and phosphoinositide dependent kinase 1. What you see as a model in the middle there is actually a demonstration of what might be the structure of phosphoinositide-dependent kinase 1, because this protein cannot be crystallized. So because it cannot be crystallized, we've had to devise different types of experiments in the cell to be able to understand how its mechanism behaves in the cell. Of course, prior to going into the cell and using different types of methodologies to be able to observe and quantify the dynamics of these proteins in the cell, many people had worked out in the test tube in classical biochemistry how these type of proteins function in relation to the phosphoinositides. So what I'm going to show you next are cartoons. And these cartoons are actually derived from the experiments that we've performed for years in the laboratory, but it's to show you how from our experiments we've reached these cartoons to be able to describe how these two proteins may behave in the cell. So what you see in the middle there is basically a shape where both, I'm going to call it PDK1, so for phosphoinositide-dependent kinase 1, and protein kinase B is PKB. So what they are, they're actually in the cell, and they have a specific dynamics together. So in the process of activating the cell in such a way that there is the formation of these polyphosphoinositides, there is a recruitment of these proteins to bind to these phosphoinositides at the plasma membrane. Now, this dynamics has been questioned of whether they recruit together or whether one of them recruits first and then binds to the other, but the event occurs in such a way that there is a change in conformation of protein kinase B upon its interaction with its master regulator PDK1. So then it has two ways to go. It goes either the left way or the right way, right, left and right side, and it can either go and phosphorylate and basically activate other proteins in a specific conformation below down here and go all the way down into the nucleus, or what it can happen is also that it forms, the PDK1 actually forms what we call a dimeric form, and it interacts with protein kinase B, which is not a dimeric form in its monomeric form, and then disassociates from the membrane and goes to activate other proteins down here. Another scenario that we have investigated is also what is its behavior as a dimer in relationship to protein kinase B, and how it might be able to disassociate from the membrane at a dimeric form. So to be able to get to this cartoon, well, we've had to be able to use our photophysics phenomena derived from Mr. Forrester, 1949, so Forrester resonance energy transfer, and just a snippet, because I don't like to compromise the physics, because then we won't understand why we've got to these cartoons. But the way that we've used basically this resonance energy transfer is to be able to measure what we call, again coming back to time, the lifetimes of these probes that we have created to be able to look at the dynamics of the change in conformation of these proteins. And how do we do this? Is by investigating the decrease in what we call the specific lifetime changes of these fluorophores that we bind to our proteins in investigation, that we basically put into the cell to be able to investigate the dynamics that that result into those cartoons that I showed you. So to show you the complexity of everything, this is what the data really looks like. And what you can see on the first panel is the labeled cells with our various probes. In this particular panel, each dot presents the different types of conformer and behavior of the proteins in the cell. And here's the quantification of the intensity and how it's recruited to the membrane in various experimental conditions. The result of this, again, is what we have recently developed, is that actually PDK1, which was thought to be monomeric and dissociate as a monomer, does dissociate. It's a dimeric form, and it dissociates as a dimer. But not only that, but it also is regulated by another type of lipid, which is another negatively charged lipid, which hadn't been shown that in cells that have a dysregulated quantity of a typical phosphoinositide and maintains this phosphoinositide at a specific level, this other negative lipid actually becomes the main source of regulating the dynamics of the recruitment and dissociation of this protein. Time passed. The mind of a man may be intent at the time of death. That is the one action. And the time of death is every moment which shall fructify in the lives of others. And do not think of the fruit of action. Fare forward. Destruction, molecular dysregulation, cancer. So after understanding all these mechanisms, we had to then go a step forward, and this is the step forward of how do we use those mechanistic events to be able to understand the behavior in dysregulation, and what methods can we develop to actually be able to quantify and go towards this element of what we call personalized medicine, to be able to determine the different activation states of biomarkers in this specific pathology. This is just to show you the complexity of the many, many types of proteins that exist, and that in one of these events, if there's even one of them that is dysregulated, you have a cascade of dysregulations, and basically the events occur in such a way that can get towards a pathology. Now, to be able to understand and to be able to quantify how this activation state and how these proteins are switched on and off in cells or in tissue, what we devised is a phenomenon, again using resonance energy transfer, but this time using antibodies, where these particular antibodies are reagents that recognize sites on our preferred protein. And in this case, again, it's an example of protein kinase B. So one antibody recognizes the on site, which is the phosphosite, and another antibody recognizes another domain of the protein, which is not the on site of the protein. And this is what we call a two-site experiment. And what this allows us to do is to reach a specificity which a one-site experiment, because of the phenomenon that antibodies may recognize different domains of proteins, one site is not sufficient for recognition, but the two sites enhances the probability of recognition and becomes more specific. We've amplified our signal to be able to have a much better signal in tissue, and the result of this, through again using resonance energy transfer, and specifically in not the domain of time, but in the domain of frequency, to measure what we call lifetimes, has resulted in understanding about quantification of different regions of tumors, of how the specific protein, in this case is breast tumor, is basically active. So this is an example of a breast tumor patient, and what you can see here is that these are the intensity images, and these intensity images portray the expression level of the protein, so the amount of protein that exists. So classically, what is quantified or what is visualized by histopathologists are the actual intensity levels. What is not quantified is the regions on these tumors which have a high activation state of this particular protein, protein kinase B. And the result of being able to understand these high activation states is to show that it is not a homogeneous phenomenon, and these events actually are quite heterogeneous. What you see here in red are the regions that are highly active in protein kinase B. So the consequence of that into the clinic is that you have what we call survival curves, and in the bottom panel of the survival curves, what you can see in blue and in red are just the expression, the amount of this protein that has been measured in patients. Now, if you take that bottom panel, what you do not see is that there is a difference between the high level and the low level expressions. So if you just do these type of experiments, you might walk away thinking that protein kinase B in breast is not a particularly valid biomarker. If you then perform the type of experiments that we have been working on, what you can see is a difference between the patients that have a high activation state, which is basically in red, and the patients that have a lower activation state. So the patients with a higher activation state of protein kinase B are indicative of a poorer prognosis. So this was quite promising, because if you just investigate levels of protein as opposed to how active they are, you might miss the process of validating the biomarkers. And again, the same is seen in a different type of cancer, which are the renal type of cancers that don't have a particular good biomarkers, so clear cell renal cell carcinoma. Again, you see the same type of phenomenon, that if you actually investigate the expression level of proteins, you do not see a difference, a significant difference, and if you investigate the activation state, you can see that protein kinase B shows a poorer prognosis. So with that in mind, we went through translation, and basically we thought that our methodology was now prepared to go and investigate more profoundly into the clinic, and even further to go and create a spin-off. And the spin-off that we created, FAST-based, standing for Threat Amplified Signal Technology for Biomarker and System Evaluation, which is based in Bilbao for the moment, brings function to pathology with a transformative platform for diagnostics and prognosis. The various types of work that we've done is on different types of predictive biomarkers in immune oncology, quantification of endogenous protein-protein interactions, and quantification of modified proteins, which is basically the phosphorylation states of the proteins. One specific example recently is that we've also developed not just what happens within the cell or within the tissue, but what happens at the surface of two cells. And we've used this methodology to actually quantify the interaction of cell surfaces. One particular example is what occurs in the immune system versus a tumor cell. And we've used this methodology to be able to understand this interaction of a specific receptor and its ligand at the surface. To do this, we've worked with, closely with pathologists in Bilbao, and because it is in the hands of the pathologist to be able to tell us where this interaction of the receptor and the ligand occur in the tissue, and for us to be able to go to these regions and to show you that it is these regions here, where you see in the arrows, that this interaction is occurring, and you can see it here in the change of the parameter that we use, the change in lifetime. These are coincident images, and the result of this is a very complicated phenomenon that occurs that the clinicians have a very difficult time to actually decipher, because the clinicians are used to one number. What we provide is not one number, but actually quantification of heterogeneity So when you look at the patients, these are lung patients, you don't have one specific number. We actually show in the different regions how they interact. So we're working on with biostatisticians to be able to reduce this in such a way that the clinicians can utilize. And quite excitingly, right now, what we've been doing with Institut Bergonier, with Jean Paloussière, is that we've also been using this methodology on patients that have gone through several cycles of chemotherapy, and they're basically don't have any other choice. So they are sent to Jean Paloussière for radiofrequency ablation. So what does that mean? Is that basically Jean, what he uses is if these are metastases in white in one side of the lung, what he does, he actually creates a lot of heat around each of these tumors, and he drives them down. So, and what we've investigating is that when he does that, what happens to the other side of the lung, and what happens to the immune system when that is done. And what we can see is that there is an event that occurs to the other side of the lung that is related to how this receptor and the ligand are interacting and how that can affect the immune system. So these are our validated biomarkers in the immune system in complex formation, so protein-protein interaction, and the modification of proteins. Time future. All manner of things shall be well when the tongues of flame are enfolded into the crowded knot of fire, and the fire and the rose are one. Reincarnation, molecular communication, unity. It is through creating the environment of common language that we can reach an entente, hopefully in our new Center Therapeutic Innovation, across the fictional borders of science, philosophy, and art. We need all three to appreciate and understand molecular evolution and translate into undoing the human malaise. The undoing of the human malaise is through our teachers, teachers of our soul. Stephen Romer. Rhythm and tone, the most important to write and maintain the poem. Jean-Paul Vacandard, healer of the soul. Peter Russell, the teacher of Plato, Republic, Symposium. Alec Linstead, the art of pronunciation and projection. Vernon David, eternally fusing the conscious with the subconscious through the cello and his unique composition. Shahid Ali, the editor that did not touch the soul of the poem, but reflected it back into the tone and the rhythm. Teachers of the mind. Colin Rosser, the first molecules of chemistry to this date. Teacher, editor of journal. Alain Boussy taught us how to understand physics with the hand and not mathematics. Dominique Pocha, the battlefield is at the bench. Continuous encouragement in all battles. Patrick Williamson, projection, future with the battlefield. Félix Gurney, to remain a Renaissance man despite all conflicts of society. Peter Parker, how to distinguish from the human psyche and just continue with science and ignore the human reaction. Alessia Alonso, how to remain a lady despite the conflicts of society in science. Eric Dufour, to understand the concepts of quantum physics in solid-state NMR The Art of the Wine, and Samuel Beckett. George Banting and Steve Ward. Humility, patience, and allowing me to come back home. Teachers of the mind and soul. M. Larijani, who has shown me order in disorder, fundamental principles of thermodynamics, patience, and battle with destiny. Mina Bahraini, her presence eternally. Cell Biophysics Laboratory, the Agora, as the Greeks say. Without every single one of them, I wouldn't be standing here. And all my colleagues from United Kingdom, United States, France, and Spain, also without every single one of them and collaborating with them, I would not be standing here. And finally, I thank Cancer Research UK for the years of funding me and being able to do most of the work I've shown you. Iker Basque, who have the intelligence to be able to fund scientists in a different way and allow them to do blue sky science. Royal Society, Medical Research Council, and the Ministerio de Economía Competitividad, Basque government, and of course fast-paced solutions. And I thank you all for your attention.
So as Head of Pharmacy and Pharmacology, it falls upon me to thank Banafshe for a quite fascinating inaugural lecture. Give you some personal thoughts while I was sitting listening to the outstanding presentation. I was really struck by the powerful images of interdisciplinary science and some really awesome images that Banafshe created and has presented to us this evening. We had a little bit of everything. We had pharmacology, biochemistry, cell biology, physics, entrepreneurship, and even a bit of poetry. So I think many people don't fully understand that Banafshe is an international, or is internationally recognised as a poet. I think that shone through aspects of this evening's presentation. But I was especially struck by the last few slides, where she's now translating her research into the clinic in the form of developing new prognostic and diagnostic markers for cancer. She clearly has a world-class reputation as an outstanding microscopist and cell biologist, and this is reflected in people in the audience this evening. We have people who come from afar, international leaders in respective fields who worked closely with Banafshe in the past. We have people streaming in from the US, Spain and France, and I think this is testament to Banafshe's international profile. I think we're really lucky to have recruited Banafshe to head up our new Centre for Therapeutic Innovation. I have no doubt that Banafshe will grow and develop the centre to be a leading entity in developing new therapeutics in the future. So please join me once again in thanking Banafshe for a quite outstanding presentation. Now what I should have said before any of that was the fact that Banafshe doesn't want to take questions at the end of this presentation. She'd rather take questions in the refreshments, which I hope will be available just outside. So if you have any questions, please save them and ask them over a refreshing glass of wine. So please make your way out into the foyer area where I believe the refreshments are.
presentation stopped it, right? Okay, I can talk now.